everyone, and welcome back to the Athenian Empower Her podcast. I'm Isabella, and today we have a wonderful guest, Marina. Thank you so much for meeting with me, and I'm so lucky to have such an amazing guest like you on today's podcast episode, and I was wondering if you could introduce yourself a little bit to our audience.
Sure. Hi, my name is Marina Sirota, and I'm a professor at UCSF, and I'm also currently the interim director of the Baker Computational Health Sciences Institute at UCSF.
Perfect. Thank you so much. And so we're going to jump right in. You have so much going on in your career, and I'm very impressed by it. But I want to know what first drew you to computational health sciences, and how did that passion of yours become, your job at UCSF?
Sure. So I actually got interested in the field of bioinformatics when I was a high school student. I didn't really know much about it. This was a while ago. I went to a public high school in San Francisco, and we had this biotech program that was led by a teacher. His name was George Kachianis. We actually reconnected recently. It's kind of crazy. But he taught this incredible course that allowed high school kids to learn a little bit more about biotechnology. And it was a lab-based course with cool experiments where we transfected genes into bacteria and made them glow in the dark, and did PCR and other things that were not usually part of the high school biology curriculum. And I loved it. I thought it was incredibly interesting and kind of sparked my curiosity. At the same time, all throughout, I was pretty good in math. So I kind of thought that I was going to go just the computer science route. And I remember sometime as a junior talking to my teacher and asking him, well, how can I bridge the fact that I'm actually pretty good in math with this field in biology, genomics specifically? And also throughout this course, I realized, well, there's some things I'm good at and some things I'm not very good at. So the lab portion, the hands-on portion of the course, I wasn't so good at. So maybe I don't have the best lab hands, but I was very curious about the subject matter.
Yeah.
So he told me, you're going to get to college, try out this field of bioinformatics. It was very new at the time. This was right around the time when the human genome was first sequenced. So there was a lot of opportunity to combine computation and biology. And that's exactly what I did. I ended up starting my career in bioinformatics as an undergraduate at Stanford.
Wow. Yeah, I love that story. I think we've seen a pattern of discovering your passion in high school and then seeing how to really make that your future career in the real world. And I think that story just pictures that perfectly. So my next question is kind of more about your work today. For listeners outside of the research world, how would you summarize the key discoveries and or takeaways from your latest Cell publication? And if you could kind of dive into your work recently.
Sure. Thanks for highlighting that. Maybe I'll step back first, and then I can dive into the
perfect. Yeah.
So in a high level, what we do in the lab and also more broadly at the Baker Computational Health Sciences Institute is we use data to ask and answer biomedical questions. And whether those questions are relating to disease diagnostics or disease therapeutics, we can use data and also different kinds of data to be able to impact human health. What do I mean by data? There's molecular data. Some examples of that are gene expression, or which genes are turned on and which genes are turned off. Or you can look at proteomics. You can look at the microbes type of measurement called the microbiome. You can look at DNA and DNA modification. So there's many different molecular technologies to characterize health and disease. And there's many labs around the world that generate these types of data. And now we also have incredible ability to characterize not only human samples, but individual cells. So we know, for instance, for certain diseases, which genes are turned on or turned off in a given cell type of interest. So it's actually fascinating.
No, that sounds really cool. Yeah.
We've gone such a far way over the last, 20 or 30 years in terms of developing these technologies. Also, because when science is funded by a government agency like NIH or many foundations, there's a mandate to share the data after it's being generated. So there's a lot of data out there in the public domain, and there are data repositories that collect these data sets. For a computational scientist, I often feel like a kid in a candy store because there's so much out there. Of course, it's messy. Maybe sometimes it's not well annotated, but there's an opportunity to use that data to understand disease mechanism. How can we predict whether somebody is more or less likely to have a certain disease?
Yeah.
That's kind of the molecular world. Now, there's a completely different type of data, and we refer to it as real world clinical data. And that's information that gets captured when somebody goes to see a doctor. That includes lab test results, diagnoses codes, medications that a patient might be on, and it's all stored in a database. It's stored in a database per hospital, but there's an opportunity to de-identify those data and do analyses on real patient data. Again, these data were not designed for research purposes, but you can do retrospective studies with those data sets as well. So really, this is the crux of what my lab does. We use both molecular and clinical data to better understand disease. And we've done that in many, many different ways. You brought up our recent paper that got published in Cell. This was work by a very talented graduate student in my lab. Her name is Yechao Li. And really, this paper is her PhD thesis. So it took a long time. It took 6 years.
Wow.
But
yeah.
I know. And this is the story behind it. Here, we were tackling Alzheimer's disease. It's a devastating disease, affects many, many people. And really, there are no treatments that are truly efficacious. What we wanted to see if we can identify already existing drugs that might help us fight Alzheimer's disease. The idea is called drug repurposing. And it's a way to speed up the drug discovery process and also make it a lot cheaper because they're already FDA approved for something else.
Got it.
So here we layer all different types of data on top of each other. We start with molecular data sets. We aggregated several data sets that measure gene expression in the brains, so this is postmortem, of patients with Alzheimer's and healthy controls. And we can compare those to see which genes are turned on and which genes are turned off. There's another database that was actually developed a long time ago, about 20 years ago. I used it in my graduate work, so it's been around, that measures which genes are affected by which drugs. So we know which genes are turned on and which genes are turned off by certain drugs.
Wow.
And we wanted to look at reversal. The hypothesis is very simple. Are there drugs that reverse those cell type specific disease signatures for Alzheimer's back to a normal state? So we ran a computational pipeline, and this is not fancy AI. This is statistics. This is kind of looking at signal versus noise, looking at reversal of these transcriptional signatures. Transcription is gene expression, which genes are turned on and which genes are turned off. And we identified 80 from a pool of 1,300 drugs. We were able to narrow down to 85 that reversed the gene expression. And we saw that 25 of them affected multiple cell types, with the idea that maybe if we were to combine several of those 25, then maybe we could identify a combination therapy, a new combination therapy for Alzheimer's. So then we went to another type of data, and this is the clinical records. Now, because these drugs are FDA approved, we can query clinical records and ask, are patients on these drugs, do they have a lower or a higher prevalence of Alzheimer's disease?
Okay, yeah.
Because we know, let's say drug X, we know who is taking, or at least who's prescribed drug X versus who is not.
Yeah.
That's what we did for those 25 drugs. By we, I mean Yechao. She led the analysis of both the molecular data and the clinical data here. So she was able to show that there were 2 drugs, and both of them are cancer drugs, that floated to the top of that reversal list. And those were irinotecan and letrozole. And they affect specific cell types. One of them affects neuronal cells, and the other one affects non-neuronal cells. So by looking at both of them, we kind of get a broader reach of potentially what's going on in the brain.
Yeah.
So then she went on, and we designed with a collaborator, his name is Yadong Huang, he's at the Gladstone Institute, animal experiments to test that in an animal model of Alzheimer's. How do these drugs work individually and how do they work in combination? And we showed that in combination, they do better. So there's memory deficits that are restored, and there are also specific Alzheimer's pathological markers in those mice that get reversed. So how cool is that? You use free data to generate a hypothesis, then you validate it in a different type of data set. The clinical records are not readily available to everybody. They're available to anybody in our institution. And then you layer on mouse data on top of that. And it's kind of the final cherry on top. What she did is she then took samples from those animals treated with the individual and the combination drugs. And she was able to show that the genes that are different in those samples, the ones from treated and untreated animals, are similar to the ones that we used on the human side for prediction to begin with. So it kind of goes full circle.
Yeah.
You start with human data, then molecular data, layer on clinical, do mouse experiments, and then tie those mouse data to the original human data that was used for prediction.
Wow.
So it's a huge study. And again, a lot of credit here goes to the student who was able to carry out this work, both on the computational side and the wet lab side, as well as all of our collaborators.
Yeah, that's so cool to me, like just hearing the behind the scenes and all the work put into this publication. I think it's so interesting. I think it's so inspiring, all of the work that you guys put in it. And I think the studying and the research behind it is just really interesting. So yeah, thank you so much. And this was recent, yeah? It was a 6-year process that ended.
Yeah, a few months ago, I think over the summer.
And
well, I will say there's still a lot to be done because mouse models are still pretty far from what happens in the human brain. So we need to do clinical trials, but this is a way that we generate hypotheses.
Yeah, I think that scientific process is just really interesting to me, and I think you put it perfectly. And that really transitions really well into my next question, which is kind of real-world application related. How do you see the research shaping the future in healthcare, improving patient outcomes in the next few decades?
Well, in my opinion, this is a good example of how you can use data to advance discovery and kind of bring these drugs closer to patients. There's still quite a bit of work that needs to be done in terms of clinical trials and making sure that these drugs are actually efficacious. But I would say this type of data-driven analysis can impact both very basic discovery science, like identifying therapeutic targets, coming up with mechanistic hypotheses, to translational work, which is what I was talking about today, to more clinical work. I mean, you can imagine using data to make workflows in the hospital more efficient, to reduce hospital readmission rates. So there's a tremendous opportunity to use computation to impact patient lives. And what my lab does is just a sliver, but there's so many people doing protein modeling, structure modeling, kind of more on the discovery side, and analysis of clinical trial data, figuring out how to get patients into clinical trials more effectively and efficiently. Again, improving outcomes like readmission rates or mortality. There's a lot of opportunities there across the board.
Yeah, I definitely see a lot of growth in the next few years for medicine. I think there's been a lot of research that I've been seeing online or on social media and of a lot of new discoveries, and I think this perfectly fits into that and where the future might take us in terms of medicine, but going back to its roots in the lab in all the testing stages.
Absolutely.
So you talked a little bit about how you worked with undergraduate students, or?
Yeah, so in my lab is diverse. We have so let me step back. UCSF is a graduate school and a medical school.
Got it.
So, for instance, the project that I talked about, Yecha was a PhD student in my lab.
Got it.
But we do have a number of undergraduate students mostly from UC Berkeley, or they come and do sometimes a post back in the lab before going on to medical school or graduate school. I also run a high school program called AI for All, with the idea of introducing kids much earlier to this concept of marrying informatics with clinical questions and bringing kind of more of the AI aspect to it. We started in 2019. The first program was in person, and then we moved to Zoom in a virtual setting, and we're very successful the first few years. Now we're seeing that the Zoom fatigue is setting in, so we need to rethink it a little bit. We took a pause this year to kind of reframe it, but hopefully we'll be back. Because I'm very passionate about getting students excited about this early. And that's one of the reasons why I decided to join you today.
Yeah, perfect. So that actually perfectly goes into my next question. I wanted to ask you about how medicine and computational health fit together, and if you could kind of explain just a little bit more about how in the future those two will go together a lot stronger.
Yeah, so I gave you an example on the therapeutic side. There's a lot to do on the diagnostic side as well. You can imagine, let's keep with the Alzheimer's kind of theme here. We had another project in the lab, and again, these are just examples from my own research, but you can expand to other disease areas and other types of data too. What we did is we used those clinical records, longitudinal clinical records, to identify Alzheimer's patients up to 7 years prior to diagnosis. So by using their longitudinal clinical records, which includes, again, diagnosis codes, lab test results, all that information, there we focused on diagnosis codes primarily. We wanted to identify patients 1 day, 1 year, 3 years, 5 years, or 7 years prior to diagnosis. And we were able to achieve accuracy of about 0.7. And this is work that came out in a journal called Nature Aging, maybe about a year and a half ago. And I'll share the link to that as well. The work was led by Alice Tang, a very talented MD PhD student in the lab. So she actually, very interestingly, she's completed her PhD, but she's continuing her medical training. So she will combine computation, kind of discovery science, and also clinical training in one. And really, it's quite amazing that we can build these predictive models. Again, we build them at UCSF. We need to validate them in other institutions to see how they hold up and how they hold up in other parts of the world, not just the US. But we can also get to the reason why. What are those features that are predictive of somebody developing Alzheimer's, and how those features are different in men and women? Your podcast is around kind of empowering women to grow and do science and do other things. Disease is very different in men and women, and that's a big focus of research in my lab. Understanding Alzheimer's is significantly more common in women, even when you adjust for the fact that women live a little longer, and we don't really know why. So in my lab, we try to understand these types of differences. How is Alzheimer's different in women than it is in men? And this study really allowed us to identify a link between osteoporosis and Alzheimer's in women, which we don't see in men at all. So that was a very detailed answer to your broad question.
No, that was perfect.
This is just another example of how you can use data, in this case, to both improve diagnostics of a very complicated and hard to diagnose condition, and also how you can understand disease heterogeneity and why the disease is different in different people.
Yeah, I think that answer was perfect, because seeing all the work behind the scenes, looking at the data, and bringing up those questions that you bring up in your lab are all really key concepts to understanding all of the work that you do. And I think that'd be really interesting to read your publication using that info, like keeping that in mind.
Absolutely.
So, I want to take a little bit of a step back, and I want to talk about a couple challenges you might have had. I want to ask what your biggest obstacle was, whether it be research or data collection or emotional, and how did you overcome that?
Oh, that's a tough question.
Yeah.
I mean, I think there are challenges along the way, different kinds of challenges. There are scientific challenges, like really hard problems. I think it's really hard for a scientist to figure out when they can't answer a certain question and when to stop a project. That's very hard, because we want to get to the answer, but sometimes we can't. And there have been a number of projects or studies where it's a negative finding. We just don't know. Maybe we don't have the data to answer a certain question. For me, also, my career path has not been linear. When I was in graduate school, I did not intend to be an academic researcher. I went to the industry first and then ended up coming back. I love what I do, but it was a very like non-linear path.
Yeah.
And I think my advice would be for young folks like you to be open to that it is a nonlinear process. And sometimes you might end up being super happy doing something that you were not thinking about at the time. So being open-minded is really, really important. And I guess more broadly, there's a big challenge. I love it all, but there's a challenge of balancing career and family. I have 3 little kids. I have a very supportive partner and family, but having a job that I love and managing both the job and family life in a way that makes me happy is not easy, but I like it. And it's really fun to figure out how these pieces kind of come together.
Yeah, that's something I've seen like across most of the women we've interviewed is they have a life outside of their work and a life in their work, and they love both, and they're finding a way to balance them. And so I think that's really like inspiring to hear from women that have been so hardworking that they still have their family and their life outside of their work, and they keep that really close to them too.
Absolutely. And I think for me, they're truly intertwined. Like during COVID, I brought all my kids to my meetings, on Zoom meetings. So I'm still in several circles. I'm known as the one with a baby in my hands, and that's okay. Everybody finds their own balance. Or actually, one of my colleagues, her name is Marilyn Ritchie. She's a phenomenal scientist and a woman role model, fantastic leader. She referred to it as work-life harmony.
Yeah.
And I really like that.
Yeah, I think that's a perfect way to put it. And that's, again, perfectly tied into my next question. I was going to ask you if you had any role models, whether it be in this field or in general, any female role models that you look up to. This podcast kind of came from the idea that we wanted to find role models we look up to, and we wanted to know their story. So if you have any role models in your field, in your life, any women that you look up to.
Yeah, I mean, I would say my role models are not only women, and I've been incredibly fortunate to have a number of mentors throughout my career. My closest mentor, Dr. Atul Butte, I did my graduate training with him, and he was actually the one who recruited me to UCSF and has shaped my career. He was an amazing scientist. Unfortunately, he passed away way too young this summer from a rare cancer, but he was a phenomenal role model, but not only a role model, he was a mentor and he was a champion. So finding people like that in your life, whether male or female, it doesn't matter, as long as you can look up to them and they can support you, I think is super important.
Yeah.
I work with a very, very talented physician scientist. Her name is Dr. Linda Giudice. She's an expert in endometriosis. We'll study endometriosis extensively in the lab. And she's also been an amazing mentor and role model to me. One of the best parts of what I do is seeing the next generation of researchers learn how to ask questions, how to work with data, and how to combine whatever their skill set is, whether it's wet lab or clinical skill sets. I've had a number of physician scientists in the lab who've learned to do these computational things. It's really inspiring, and I think it keeps me going.
Yeah, I love that. Thank you so much. That's all the questions I have, but is there anything you want to add, anything you want to touch base on?
I guess, well, thank you for having me. It was really fun to chat with you. I urge the listeners of your podcast to be open-minded, to think about what really inspires them, what really inspires you, what kind of questions are exciting to you. Find a problem that pisses you off or something that you feel like would be impactful. And even if it's hard, go for it, try it, and maybe it'll work out. And if it doesn't, that's okay too. You try something else. So I really enjoy this kind of intersection of the two fields that I've dedicated my career to. And it's been a lot of fun. So I wish you the same.
Amazing. Thank you so much. Thank you to everyone who tuned in. Make sure to check out our YouTube, Spotify, and Instagram, and we'll see you next month for next month's episode.